Alright, so let's go ahead and do our new words. Now we're learning about the cell cycle today. So now the first word you need to write down is interphase. What is interphase? Interphase is the longest phase of the cell cycle. 90% of the time will be in interphase. 90% of the time will be in interphase. It contains G1, S, and G2. Alright, so it's going to contain G1, S, and G2. It has all three of those parts. All three of those parts are part of interphase. If you see there behind me, you see all that green area? That's all interphase. That's containing G1, S, and G2. All that is part of interphase. The longest phase, 90% of the time spent in that. Interphase. Alright, so now the next word is G1. G1 is the growth phase. For G1, put the growth phase. G1, growth phase. So now the second word is S phase. For S phase, put this down. DNA replication. For S phase, put DNA replication. The DNA is being copied. DNA replication. DNA is being copied. S phase. Alright, S phase, DNA replication. The next one is G2. For G2, put down second growth phase. Second growth phase. G2 is the second growth phase. Getting ready for mitosis. Getting ready for mitosis. So again, G2, second growth phase, getting ready for mitosis. That's G2. Alright, so now we're on the M phase. M phase. For M phase, put this down. Mitosis phase. Mitosis phase. M phase is the mitosis phase. PMAT. Write down PMAT. PMAT. M phase, mitosis phase, PMAT. Which would be prophase, metaphase, anaphase, telophase, which are the words below it. PMAT contains PMAT. So M phase, mitosis phase, PMAT contains PMAT. Prophase, metaphase, anaphase, telophase. Alright, so mitosis is when you're making body cells, somatic. So write this down. Mitosis, put down body cells, mitose. Okay, mitose. Your toes are your body cells. So mitosis, think of mitose. Body cells. Somatic. Somatic is like saying body cells. Somatic. So write that down. Somatic. Body cells. Somatic. Put this down. 2N. The number 2 and then the letter N. 2N. The 2N is because you have 2 sets of chromosomes. That's why it's 2N, because you got 2 sets of chromosomes. Genetic information identical. Genetic information is identical. 46 chromosomes for humans. 46 chromosomes for humans. Alright, so whenever you make a mitosis, a body cell, if you're a human and you're doing mitosis, you're making a body cell, it's going to make 46. They're going to each of those cells are going to have 46 chromosomes in them, because humans have 46 chromosomes. Alright, so 46 in humans. Diploid. Diploid cells. Diploid, put diploid daughter cells. Diploid daughter cells. Okay? So, body cells, somatic. 2N. Diploid daughter cells. Identical genetic information. 46 in humans. 46 in humans. That's mitosis. And then last thing to put down is PMAT. PMAT. Mitosis has 4 main phases, which is PMAT, prophase, metaphase, anaphase, telophase. Alright, mitosis, 4 main phases, which is prophase, metaphase, anaphase, telophase, PMAT. Mitosis. All right, so the next word is prophase. With prophase, what's going to happen is the nucleic envelope or slash nucleic membrane. So it could either say, you could say nucleic envelope or you could say nucleic membrane. That's why we're going to put nucleic envelope slash membrane begins to disappear, okay, begins to dissolve. Chromosomes condense. Chromosomes condense. All right. Now we're just trying to get close together. And then you could actually see them. Chromosomes condense. You can see the X's. All right, put that down. Chromosomes condense. You can see the X's. Write it down because you'll see little X's, and you'll see that the nucleic envelope starts to dissolve, and then you could put looks like dotted line. Looks like a dotted line. So nucleic envelope starts to dissolve. It's going to look like a dotted line, like you see there behind me. And then the chromosomes condense. They start to look like and become visible. Chromosomes condense and become visible. They start to look like X's. So one last time. Prophase. Chromosomes condense and become visible. They look like X's. And then the nucleic envelope slash membrane dissolves. Starts to look like a dotted line. All right, and that is prophase. All right, so the next word is metaphase. For metaphase, you can put chromosomes line up along the middle. Equatorial plate. Chromosomes line up along the middle equatorial plate. The X's look like they're in a straight line. The X's are the chromosomes, okay? The X's slash chromosomes get in a straight line, like you see there behind me. You can say spindle fibers come out and attach to the centromeres. Spindle fibers come out and attach to the centromeres of the chromosomes. Put this down. Meta, middle. Meta, middle. All right, so now we're on anaphase. For anaphase, put chromosomes are being pulled apart or away. Chromosomes are being pulled apart or away. I like to think of it as looking like little sideways V's, or you could think of it like sideways capital A's. See how they're like that way? So that would be anaphase. Chromosomes are being pulled away or apart. You could put ana away. Ana away. All right, so now we are on telophase. For telophase, you can put 2 new cells begin to form. So for telophase, you could put 2 new cells begin to form. 2 new nucleic envelopes begin to form. You could put looks like a telephone, like an old telephone. Looks like an old telephone. Some people say it looks like breasts. So if that helps you remember it. So you could say it looks like a telephone or it looks like breasts. Easiest way to remember is 2 new cells begin to form. And I would say telophase, telephone, and I try to picture it to look more like an old telephone where you have the earpiece and the speaking piece in 2 different spots. All right, so this next one is cytokinesis. So with cytokinesis, what you put down is pinching off of the cell membranes, pinching off of the cell membranes. Nuclear envelopes have already formed around the nucleus, and pinching off of the cell membrane. All right, cytokinesis. You could also put this down, help you remember cytokinesis. Cytokinesis, 2 pieces. Cytokinesis, 2 pieces. All right, you don't have 2 cells already fully done, but you're pretty much, it's just pinching it off to end it. All right, because if you had 2, then you'd already be in, there'd be 2 separate cells. You'd be done. So you're still in a phase here, cytokinesis, but you're like almost done. It's just pinching off the cell membranes and it's over. All right, so cytokinesis, 2 pieces. All right, so now the next word is chromatid. Chromatid is each strand of the chromosome. All right, so if the chromosome is the X's, you see those over here, chromosomes? All right, the chromatid is each strand over here. You see how one of those strands is a chromatid, all right? Where when you have the two of them, you see how there's 2 here? Here's one chromatid, here's another chromatid. You put both of those strands together, now you got a chromosome, all right? So chromatid, a strand, a single strand from a chromosome. And if we're saying the chromosomes that they look like X's, the chromatid would be like one of the lines that makes the X's. All right. All right. So next word is spindle fiber. Now I could say filaments that form mitotic spindle, made of microtubules, but really that's not going to help you much, because that doesn't really help. So just put this down for spindle fibers. It's gonna seem silly, but really all you're gonna probably have to do on a test is identify it. So if you look at the picture there, if it said, hey, where are the spindle fibers? You see it's labeled there behind me, but you could just look for the little spider web looking thing. So for a spindle fiber, just put, looks like a spider web rope looking thing that grabs onto the centromeres, the center of the chromosomes. All right. Or again, you